Okay, welcome. Thanks for tuning in. Today I want to talk to you about something that I believe is really, really important. And I think that while some of this information is out there on the internet, some of it may not be. You may already understand some of this. You may not understand any of this. Whatever the situation is, I think you owe it to yourself and to everybody you love to make sure that you get them to have a look at this information. Whether it's through this lecture that I'm giving or through other lectures given by others along similar lines. The main point is that you get hold of this information and understand this, because this is, as I say, really, really important. Okay, so let's talk about cholesterol. Cholesterol. We've all heard about cholesterol. I don't know how many times. It's in the news. It's on TV just about every night with these margarines and things that you can get that supposedly lower your cholesterol, and that this is supposed to be a good thing and all that kind of business. There's no doubt out that if you've been alive for the last 20 years, you've heard all about cholesterol. And you've probably also heard that there is good cholesterol and there is bad cholesterol. So let's put that one to bed right here and right now. On the screen is a model for the molecule cholesterol. Okay, you can look in any biochemistry textbook you like. You can look it up online. There is only one cholesterol molecule. That's it. That's what it looks like. It is neither good nor bad in the sense that it's being portrayed I guess out there in the media and with those with vested financial interests in this area. It's a molecule that exists in your body because, as we're moving through the lecture, you'll get to see there's a whole bunch of genes that encode for its transcription. Now those genes exist because they have survived 3.5 to 3.8 billion years of natural selection. They're there for a reason. As is cholesterol, it serves very, very useful purposes. In fact, without cholesterol in your body, you will die very, very quickly. It is absolutely vital in your body. And in fact, this whole idea that we can have too much cholesterol in our blood only really applies to people who have a specific genetic knockout. That means that they don't have a receptor for cholesterol transport, which means they get very, very high blood cholesterol levels. And that expresses itself in about 1 in a million in the population. So, the chances of you being one of those people is, as I say, about 1 in a million, and you'd know about it because you'd have been diagnosed as a very young child. And those people don't generally live long lives. People without that problem have a level of cholesterol in their blood that's exactly what it should be, and we'll get to that as we move through the lecture. So, moving on from there, let's talk about what cholesterol is. It's a waxy form of a high molecular weight alcohol, actually, is how it's categorized by the chemists. A lot of them call it a lipid. They call it a lipid because it's not water soluble. Lipids are generally thought of by most people as kind of fats, but you need to understand that cholesterol is not a fat. It's not a form of fat. It's a form of alcohol. It's just a high molecular weight alcohol that has a sort of waxy physical presentation. The vast majority of the cholesterol in your body is synthesized by your body. I'll show you exactly how that occurs in a bit as we move on through the lecture. Dietary intakes make no real difference to how much cholesterol there is in your body, and we'll talk a little bit about that as we move through as well. What you need to understand right up front is that 30% of every cell membrane and every cell in your body by weight, 30% of those cell membranes is cholesterol. Okay, so again, without cholesterol you cannot make cell membranes. Without cell membranes you can't have cells. Without cells you cannot live. So right then and there you can see, okay, cholesterol exists in our body for a reason. It has a role to play. That's one of the many roles it plays. That's one of the perhaps most important ones. Moving on from there, both estrogen and testosterone, which are the male and female sex hormones, if you like, or gender hormones, They're biosynthesized directly from cholesterol as the precursor, and there are just so many myriad issues that can occur in your body if you mess around with your ability to make estrogen or testosterone. In the case of estrogen in women, lowered estrogen will lead very directly to a loss of bone mineral density, osteoporosis, which can lead to very serious secondary issues from that, including hip fractures and blood clots and all the kind of nasty stuff that you really don't even want to be looking at getting into. In men, testosterone, yep, without that, you're going to have trouble having muscle mass, you're going to have a reduced functional strength, and you're going to have a reduced bone mass as a function of that via Wolff's Law. There's a whole bunch of other stuff that's going to go on there that's not going to be real good if you can't produce testosterone. So, yeah, cholesterol is the precursor for that, so there's another very important role that cholesterol has in your body. Myelin sheaths. Okay, myelin sheaths are kind of like the wrapping around all the different neurons, all the different nerve cells in your brain and in your peripheral nervous system as well. Myelin is the stuff that means that you are able to transmit nerve signals around efficiently and quickly around your body, and yep, without cholesterol you can't actually insulate those neurons, and then you get all sorts of short circuits and misfiring problems, and yeah, it's just not, you don't want to be going there. Again, that's cholesterol, folks. And also downstream of cholesterol you're going to find things like vitamin D. Now vitamin D, there are several different forms of vitamin D, but its overall roles are many and varied in the body. Let's just go to one very simple, very basic example. I'm going to show you some enzymes in a minute, and I'm going to show you a very few enzymes. There are thousands and thousands and thousands of enzymes in your body, tens of thousands actually, and fully 1/3 of those enzymes are activated by vitamin D and deactivated by lack of vitamin D. You knock out cholesterol, you knock out vitamin D, you knock out your ability to undertake the chemical reactions that your body needs to undertake in order to keep yourself healthy. Not a good thing. Also, bile comes from cholesterol, and bile is very, very important in your ability to be able to take in and use fats in your body. So this is like a brief sort of overview of some of the things that cholesterol does. So hopefully what you're getting already from this lecture is that cholesterol is not your enemy. It's not a bad guy. It's not something we need to lower or get rid of, or it's not something that causes us a problem. The whole idea that it does was created by people with a vested financial interest in suggesting that it does, and by the end of this lecture you will see why none of those arguments hold any water whatsoever. Some other facts about cholesterol that you may or may not know or find interesting. Because of its waxy nature, cholesterol is hydrophobic, meaning it doesn't mix with water. It will float on water. Your blood is based on water, so to get cholesterol transported around your body in the blood, we basically need to come up with some packaging to package this stuff that won't mix with water up inside some, what they call lipoproteins, or phospholipoproteins if you like, that are kind of like balloons. I'll show you what they look like in a minute, and they encase the cholesterol along with other fats and lipids that don't mix with water, and they are packages that can move around in the blood. They will dissolve in the blood, they will be in the blood, and they can get the cholesterol to where it needs to be. There's a whole bunch of different sort of packet sizes, and they have different densities in terms of the protein content and the fat content, etc., etc. And it's these different proteins that determine where that particular package of cholesterol and other lipids is going to end up being delivered to within your body. Each one of them has a different receptor which exists in different parts of your body, where the different cells exist that need the different packages to deliver the different payloads of cholesterol and lipids and things. Okay, you need to know that these lipoproteins are expressed, they are synthesized by your body. There is DNA that encodes for the genes, there are DNA that encodes for the enzymes that facilitate the proteins being put together, and the rate at which these proteins are made is dependent entirely on your need to transport and move various forms of cholesterol around your body. What you eat doesn't really seem to have a great deal of impact on the expression of those proteins. So again, this is not a dietary issue. If you could meaningfully affect your cholesterol by changing your diet, then we wouldn't need a drug that does it. Okay? Actually, as it turns out, we don't need this drug anyway, but we'll get to that. Okay, so here's a visual, if you like. Here's what the lipoproteins look like. There are various different classes. They come in different sizes. The larger they are, the lower their density. So the lowest density lipoprotein is called a chylomicron, or chylomicron if you like. Those little sort of orangey, yellowy, reddy sort of spheres that you can see within the structure they're the graphical representation, I guess, of the water-loving or hydrophilic phosphate end of a phospholipid. On the inside, hidden from our view there's a couple of tails of fatty acids, and these there are two layers of these, one facing inwards, one facing outwards, basically, which allows the inside of the sphere to contain the cholesterol and the triglycerides and other fats and other things that won't dissolve in water. And the kind of whitish or off-whitish stringy thing you can see is the particular protein that holds that structure together, that holds that sphere together, I guess, that encodes for it, and it's that protein that the different receptors on the different areas of your body will recognize and grab those particular packages as and when they need them. So the next one down here is what's called a very low density lipoprotein, or VLDL. Different protein, different size. Down here we've got an IDL or intermediate size one. Again, a different protein that encodes for that. LDL, this is the one that you've probably heard of as the bad cholesterol. Okay, we'll get to that soon. Again, we've talked about the molecule cholesterol itself. Cholesterol is not bad. Cholesterol is actually vital. So when they're talking about bad cholesterol, what they're actually talking about is a bad form of the lipoprotein. What you also need to understand, I'll cover this more detail soon, is that it's not the native form of LDL that's a problem. It's only when that LDL becomes oxidized and damaged that it becomes any kind of problem. We'll get to that. Excuse me. And then the smallest category you can see there is called an HDL, or high density lipoprotein. That's the one that they usually refer to as the good cholesterol, and I'll tell you why it's referred to as the good cholesterol later, and then I'll also tell you why that's not so either. Yeah, it's a bunch of misdirects, it's a bunch of nonsense put out there by scientists, I guess, who have been, shall we say, encouraged to spin the science in a way that suits those with a vested financial interest in spinning it that way. But I have no axe to grind in terms of financially. I'm just here to tell you what the science actually tells us. So we'll get to all of that as we move through it. But anyway, for what it's worth, that's kind of what they look like. Or that's a representation of what they look like, to be fair. Little spheres, little bubbles, if you like, which will move through water without separating from it, inside which you find the fats, and it's all wrapped up by a sort of stringy protein structure that holds it all together. Okay. Here's kind of like an analysis of the makeup of the different classes of lipoproteins, from the chylomicron right through to the smallest or high density lipoproteins. It kind of gives you an idea of the size and the makeup of those in terms of the red slices are how much cholesterol you have in them, the green obviously how much protein, and then phospholipids, That's the orange bally structure thing that you could see on the previous slide. And then triglycerides are, your fats that you usually incoming from your dietary intake. Okay, so really that's all we need to say about that. Pretty much it just shows you that as the amount of lipoproteins and the amount of cholesterol inside these lipoproteins changes, there's also a change of protein structure that holds it together, which encodes it to be delivered to different areas for different purposes. Basically, the take-home message, if there's any on this, is that each one of these classes of lipoproteins exists because there are genes that encode for its existence, and those genes exist because those genes have survived natural selection for somewhere between 3.5 to 3.8 billion years. They're there for a reason. They all do a job. Okay. Here's a sort of scheme that kind of shows the movement and fate of cholesterol in the body in a sort of simplified way, if you like. If we start right over on the left, we can see a small representation of an intestine there, and what you'll see is some little yellow spheres with a little C beside them, and those little yellow spheres, you'll see them entering an intestinal cell on the wall of the intestine. So that would be cholesterol in the food that you've eaten, and it gets absorbed through the wall of the intestine into this intestinal cell, where if you need cholesterol in your body elsewhere, there will be some chylomicrons waiting for that bountiful cholesterol, that supply of cholesterol. So that will go into the hold of that chylomicron ship, if you like, ready for transport out into the bloodstream to go to the cells of the body. Now, as I said before, most of the cholesterol in your body, in your bloodstream, in your cells, anywhere else in your body at any given time is generated directly by your body from other food sources. It's not that you have a lot of cholesterol because you've eaten a lot of cholesterol. If you have a lot of cholesterol, it's because your body has synthesized a lot of cholesterol from other things. However, if you eat cholesterol, your body can absorb cholesterol, is what we're saying. Alright. What you'll also see right underneath that is another receptor called ABCG5/G8. Now what that thing does is, if there's more cholesterol in the cells of your intestinal wall than you need right now, it spits the cholesterol back out into the intestine. Where does it go from there? Basically, you shit it out. So to say that eating a lot of cholesterol is problematic right there is a ridiculous comment, because your body is designed to absorb and use what it needs and what it can and get rid of what it can't. There it is right there. You eat too much cholesterol, you shit it out. Okay. If you need it, along with some fatty acids, which are shown there in red, they get mixed together and put inside a chylomicron, which has a protein, as you can see, they're called B48. And it goes into the bloodstream, whereby the fatty acids can be delivered to your fat cells directly if you want to store some fat. They can also be delivered to other tissues where that fat is going to be oxidized and used for energy, if that's what's required at the time. Once the fatty acids have been dumped off, you've got what's called a CMR, or a chylomicron remnant, if you like. That is recognized by the LRP1 receptor, and those CMRs will then enter the liver, where basically they can then be changed into very low density lipoproteins. And again, there's a system there by which there's a huge amount of storage of cholesterol and triglycerides in your liver, and they can be drawn upon or added to, much like a bank account really. If there's more money coming in than is being spent, then the balance goes up, and so long as there's some money there at all. If you spend more than is coming in, then the balance goes down. Obviously with things like triglycerides and cholesterol, there's no overdraft. There's however much there is, but the adage, I guess, is still good. From there, you've got your intermediates and your low density lipoproteins, and basically the determinant as to which one of those it is again which protein is encoding around the outside of it, and how much cholesterol and triglyceride is inside it. So these particles get progressively smaller and more dense in terms of the protein ratio in them, as the cholesterol is sequestered and used by various tissues around your body as and when it's needed. The only other thing there that I would point out to you is that the other class there of lipoproteins, the HDL, shown sort of in a blue-green there with the A1 pink protein slice there. That's kind of a special class of lipoprotein because its major role is actually in scavenging up cholesterol from around the various different sources, from peripheral cells that are not needing it, that are exuding it, for example, or if it accumulates where it's not needed anymore, for example, in the walls of your vasculature or something like that. This HDL will collect up that cholesterol and deliver it back to the liver. So basically it's a scavenger. It's called reverse transport. So its role is not so much in delivery, but in scavenging up, cleaning up, and taking the cholesterol back to the liver. Okay, so there's basically a scheme on that for what it's worth. So some salient points to that. As I said, if you eat too much cholesterol, you shit it back out. Okay. All the different forms of the lipoproteins are involved in delivering specific cholesterol, triglyceride mixtures to specific cells, depending on what protein is on them, because the cells are programmed to lock onto and to sort of draw on one of those particular classes of lipoproteins. So it makes sure that the right stuff gets to the right place at the right time. They all exist for a reason. That's why they're there. Yeah, there's a bunch of different roles that they play, including one that collects up cholesterol and takes it back to the liver. So they're the salient points there. So let's talk now, moving on to the disease which is known as atherosclerosis. Basically how it progresses is that the normal artery, as you can see at the top there, has nice, smooth, slick vascular walls. The blood flows through there with an amount of turbulence still, but in a reasonably sort of smooth sort of a fashion, although it's pulsatile. And then when the disease atherosclerosis starts to occur, what happens is that there's this build-up of tissue. It's actually mostly scar tissue, I'll cover that soon, which starts to occlude or invade into the inside, or the lumen is the technical word for the inside of the artery, and that starts to block off the blood flow, cause more turbulence, cause more problems, which then eventually in a very serious case can lead to that particular artery becoming completely blocked off. A clot will form, and then usually at some point that clot will break off, float around in the bloodstream somewhere until it lodges in a small enough artery to block that artery off and cause a problem. If that's in a heart, that's called a heart attack. If that's in your brain, it's a form of stroke. If it's in your lungs, that'll cause another sort of problem there as well. If it's in a muscle, you could get some muscle damage there. The process of heart disease, though, is one of these blockages blocking the artery directly in your heart, meaning that your heart tissue dies, which is obviously not a good thing. It's important to understand that atherosclerotic lesions, these plaques that we're talking about, do not occur in the veins. They only occur in the high pressure side of the vasculature. They only occur in the arteries. So that's the first point to keep in mind about causality. We have been told for years and years and years that the cause of atherosclerosis, or one of the root causes of atherosclerosis, is cholesterol in the blood, and too much cholesterol in the blood. Now, the cholesterol level in the veins is going to be roughly the same as the cholesterol level in the arteries. It's the same blood. It's just returning back to the heart. So if that were remotely a sensible argument, then atherosclerosis would be occurring in veins as well as in arteries. It doesn't. So there needs to be something else occurring, and what that thing is that you need the pressure of the high pressure side of the vasculature to drive cholesterol into the gap junctions in between the cells in the walls of those arteries. In a normal healthy person, that's not going to happen. So while cholesterol may be involved, a certain form of cholesterol specifically may be involved in the disease process, it's not a cause of the disease process. To sort out the cause of the disease process, we need to sort out the thing that's initiating the process in the first place. And I'll talk to you about what that is very soon. Here's a sort of more or less simplistic diagram, if you like, which shows more about the process. Where it says lumen there at the top left, that means the inside of the artery concerned. You can see some red blood cells and some white blood cells, which are actually shown as a sort of purpley, sky bluey colour there. And they're moving around, and you'll also see some of those friendly yellow cholesterol globs. Those yellow globs in this diagram represent the LDL, the low density lipoprotein subfraction. And what you can see is that under pressure, when there are gaps between the cells that line the inside of the artery, those little lipoprotein molecules can get shoved into those gaps. Okay. Question 1. Why are there gaps there? Okay. Here's the answer. They are caused by dysfunction at the level of those cells inside that particular artery, and the most obvious common cause of those problems is systemic inflammation caused by, most commonly, a poor diet, intemperate habits, lack of appropriate exercise and sunlight, etc, etc, etc. Smoking's another big one, but the single biggest cause of systemic inflammation is a poor diet. Anyhow, when that's been happening, we're going to get these gaps, which are going to be caused by this huge amount of inflammation and cellular dysfunction on the vascular epithelial cells. This cholesterol is going to be driven into these gaps and become stuck in there. When that occurs, over time, those LDL particles become oxidized, which they wouldn't have done had they been free to move around back through the blood and be delivered back to the liver and be used again. Basically, they're kind of rammed in there, they're stuck in there, and they go off. Okay, they become oxidized. Then your body recognizes them as an invading protein. They're not your natural protein anymore, and so the white cells will jump in there, as you can see at number 3, and they will start to gobble up these oxidized LDL particles, and then they turn into these things called foam cells, which you'll see there at number 5, and that's where the plaque starts to develop. The whole area is invaded by new scar tissue to encapsulate and encase that whole problem so that it doesn't become a systemic problem. That's why your body's doing it. And so you just get this runaway situation where you just get this huge development of scar tissue, which then forms a bulge, which then blocks off the artery, and then we get the blood clots and all the problems that we were talking about in the previous slide. Probably should have given you a warning about that, but there you go. Kind of looks like pizza, doesn't it? It's with too much cheese. That's the inside of the main artery that comes out of the top of your heart. That's been split open along its length and opened up so that you can have a look at these damaged areas, these atherosclerotic lesions that you can see. Yep, so that's kind of how the disease looks and how it manifests itself. So as you can see in one of these areas, if one of these scabby areas breaks off, that blood clot can then move around through that artery and it will work its way to a smaller artery where it will jam in the heart, and if that's where it ends up, and cause, for example, a heart attack. So let's talk about some atherosclerosis facts and findings. Low density lipoprotein cholesterol, LDLC, is indeed associated with atherosclerosis. Okay, so whenever you've got atherosclerotic lesions, you will find that there's low density lipoprotein present in those lesions, and I'll talk to you about how much you'll see there soon. But what we're saying is, yes, it's there. However, every time there's a forest fire, you're going to find trees. But it would be absolutely ridiculous to say that the cause of forest fires is trees, wouldn't it? That's not how things work, okay? It's important to remember that only when that LDLC can invade the intima and the media, which is the tissue inside the lining of the arteries, only when that LDL can get jammed in there, when there's gaps in those junctions, that the disease can only progress under those circumstances. If those gap junctions are healthy and tight, those cells are healthy, there's no runaway systemic inflammation going on, atherosclerosis cannot occur and does not occur, okay, irrespective of how much cholesterol you have in your blood. Okay, so again, cholesterol is not the cause. Okay, and once the cholesterol is jammed in there, it becomes oxidized, and only when it is oxidized will it become a problem. Only then will your white blood cells attack it. Only then will that system of inflammation become runaway and that scar tissue get laid down and those lesions develop. Okay, so I hope all of that's fairly clear. A lot of people will talk about the fatty plaques, the fatty atherosclerotic plaques. Okay, let's be clear about this. They are not fatty. They are largely fibrous. They are largely scar tissue in their makeup. We can cut these lesions out of people who have passed away. We can put them under a microscope and we can look at these things. They are fibrous. They are not fatty. It's a ridiculous comment to say they are fatty plaques. It's spin, again, that is put there by people with a vested interest in getting you to believe that fat is a problem and cholesterol is a problem. Okay? We need to understand that the cholesterol content of these plaques is somewhere between 20 and 75 milligrams per gram, which accounts by weight to somewhere between 0.2 and 0.75% of the entire atherosclerotic lesion. Okay, this is not a primary constituent of these lesions. It's a very, very small amount of stuff there. The overall fat contents in the plaques is indeed stunningly low. It's 0.05% by weight. Of the fat that is in these plaques, fully 70 to 85% of that fat is monounsaturated fatty acids and polyunsaturated fatty acids, in other words, heart healthy oils, the stuff that we are sold and told is good for us. Okay, around about 15 to 30% of the fat is the saturated fat. So let's have a look at that. Yep, basically what I've just been saying, that shows you those ratios there as to what's in those lesions once they've been dissected out of people who have passed away from atherosclerosis or whatever cause, and we've been able to work out what's in these lesions. Here's a makeup of some of the fats in there: saturated, monounsaturated, and polyunsaturated. W6 means the omega-6 oils, and W3 is the omega-3 oils. Okay, so that's what's in there. So when you hear the term fatty lesions or fatty plaques inside the arteries, nonsense. Not fatty at all. Hardly any fat there whatsoever. Artery clogging cholesterol? Nope. There's hardly any cholesterol there. It's scar tissue. Artery clogging saturated fat? Again, nope. That doesn't add up either. Just a slide for what it's worth. Have a look there. Probably pause this at some time and just have a read through some of those if you want to work it out. The reason I had the slide in there, when I was talking to my students, I've been a lifelong advocate of low carbohydrate, high fat, moderate protein nutrition throughout the lifespan. And this was just to give them an idea of some of the fat profiles and some of the fatty foods so that they could make good choices in terms of that. This is organised in such a way in terms of the makeup of the fatty acids in there that you would read it from top to bottom, the bottom being the best oils that you can choose or fats that you can choose, and the top being the absolute worst. So if you read through some of that might be a shocker to some of you. One of the main reasons for actually avoiding the heart healthy oils, the omega-6 oils in particular, is that these have been shown to be very pro-inflammatory. They cause a systemic inflammation. They cause irritation of the cells inside your arteries. That's what causes the gap junctions to open up. That's what allows the cholesterol to get jammed in there. That's what starts the problem. So these are, among other causes, the intake of too much carbohydrates, another one that causes massive inflammation, but we'll get to that in other lectures. So anyway, that's why that slide was there. Have a look in your own time. So the apparent actual causes of atherosclerosis are hypertensive, in other words, high blood pressure, because we spoke earlier about that. We need high blood pressure. We need to be able to drive that cholesterol into those gap junctions. So hypertensive and other damage and inflammation to the epithelial cells. Okay, so examples I've just been talking about: too much carbohydrate, too much healthy heart oil, not enough healthy saturated fats, smoking, either insufficient or too much exercise or inappropriate exercise modality, smoking, drinking, you name it, all those kind of things that can have an effect on our level of systemic inflammation. Hey, even environmental toxins and poisons that float around, pollution and those kind of problems can have an effect. Another possible cause of damaged epithelial cells can be viral or bacterial damage. Again, so that it may well be that there's a disease process there that can be communicable that can start off atherosclerosis as well. So that's just a point there out of interest. So as I said before, when you've got an occlusion in or a blockage in an artery inside the heart, that's what we call a heart attack. If it's in the brain, that's one of the various forms of stroke, those kind of things. So that's just a graphical demonstration of that for you, really, for what it's worth. Okay, so probably everybody that's been alive any time in the last 20-30 years at least has heard that saturated fat will clog your arteries. Well, no, it won't, because we've already had a look at what's in these plaques, and there's hardly any saturated fat there at all. And that if you eat saturated fat, you're asking for heart disease, and you need to replace that with all sorts of healthy whole grains, which is an absolute load of nonsense, because it's the whole grains and the avoidance of saturated fat that's driving the inflammation that's causing the heart disease in the first place. But here's a meta-analysis, which is a synthesis of a bunch of studies done previous to this study. And what these authors have done, they've used a thing called a meta-analysis, which is a statistical slice and dice methodology, which allows them to put the results of a bunch of different studies together and make a much bigger study, which has obviously much more statistical power and is therefore much more reliable. And what they did is they had a look at the association between the amount of saturated fat in the diet and deaths from either coronary heart disease or cerebrovascular disease, so strokes and heart attacks, basically. The things that they tell us saturated fat will predispose us for. Okay, so what they've got there, if we look under results, is that they have followed a total of 347,747 subjects over a number of years, which gives us a number of person years of follow-up in the sort of hundreds of thousands. So we've got this huge sample size there, really. And what they have concluded is that this meta-analysis of prospective epidemiologic studies showed that there is no significant evidence for concluding that dietary saturated fat is associated with an increased risk of coronary heart disease or cerebrovascular disease. Okay? There is no evidence that people who eat more saturated fat have more health problems of this kind. Okay? So it's an idea that's had its time. It's an idea that needs to go. Okay? The avoidance of saturated fat is not indicated. It should not be done because there is significant evidence to say that the stuff we're replacing the saturated fat with in our diets is causing us a problem. And they are carbohydrates and most notably polyunsaturated fatty acids. Having said that about this study here, this was a 2010 study, there's a thing called publication bias. And what publication bias is that in science there's this ridiculous idea that if something is 95% likely to be a real thing statistically, we publish it. And if it's 94.9% likely to be a real thing, we ignore it and say it's not real. So it's a threshold test. Bigger and bigger and bigger studies have more and more and more probability of being published because they are bigger studies and have more power. Smaller and smaller studies have less likelihood of getting published, especially if they don't find 95% probability one way or the other, and so they end up in someone's bottom drawer, never to be seen. So what does that look like? Let's analyze the data that comes from this study that I'm talking about here. You get this thing called a funnel plot. Sometimes it's called other things. I like to call it a funnel plot. And it plots the results of all the different studies that have been used in the synthesis of this overall meta-analysis. The further up this triangle shape that you go, the smaller the error of that study is statistically, because they are bigger studies. They have larger sample sizes, more people involved in those studies. There's a thing called a standard error of a finding, and the standard error is the standard deviation of everybody in that study divided by the square root of the number of people that are in that study. So what that says is the bigger your study is, the more likely it is to be representative of the real world finding in terms of that particular observation. So you go lower down and you'll find that studies will splay out further and further away from the 1 risk ratio, which means there's no change in risk. But often what you find in these studies is exactly what we find here, and that if you look at the bottom left hand of that funnel plot, you'll see that there's no studies in there. What that means is that only the smaller studies that did find a 95% this is a real thing, and yes, saturated fat was associated with more heart disease and more cerebrovascular disease, they're the ones that got published. They're the ones that didn't end up in someone's bottom drawer. So what they do is they actually tend to drag the end result of the study, which in this case was around about 1, no change in risk. It tends to drag it to the right. In other words, towards this study being more likely to find that saturated fat was associated with increased risk than is the real situation. I hope that was understandable. What I'm saying is this study found there was no increased risk, despite the fact that it was always biased towards finding that there was a risk. So in fact, while it says there's no risk, what I'm suggesting is that there is actually a possibility that eating more saturated fat and less other stuff is actually cardioprotective. More studies need to be done. Okay, so again, atherosclerosis has been blamed largely for many years now on cholesterol, and mainly they've now said, it's actually the low density cholesterol that's the problem. Okay, well, what are we looking at here? We are looking at a chart which shows the blood cholesterol level in the top one, the red histogram. This is the blood low density low density lipoprotein cholesterol level in patients at the time that they were admitted to hospital with a heart attack. Okay? Now, we are told that increasing the concentration of LDL in your blood will increase your risk of having a heart attack. Okay, if that were true, what would we expect to see here? Well, we would expect to see the number of people presenting with a heart attack going up and up and up and up for every increase in blood cholesterol that we've measured. Is that what we're seeing here? No. No, it isn't. We are seeing a normal distribution. Slightly skewed, sure, but a normal distribution. What that means, if you look at this chart, is that there were far, far, far fewer patients presenting to hospital with a heart attack who had a blood cholesterol LDL level of 220 milligrams per deciliter of blood than those who had 90 or 100. Far, far fewer. That's not what we'd see if increasing the cholesterol further and further and further increased the risk further and further and further. End of discussion. This is nonsense. Okay. So, low density lipoprotein cholesterol is not bad cholesterol. Let's have a look at whether HDL is good cholesterol. Okay? Same argument applies. What would we expect to see if that was the case? Well, for every decrease in HDL cholesterol as measured in these patients that have presented with a heart attack, we would see an increase in the heart attack. So it would be an inverse relationship to the red one. Again, it's a normal distribution. We're not seeing that. HDL cholesterol is not good cholesterol. It's just another form of cholesterol. They're all good. Actually, all the forms of cholesterol, lipoproteins, are all good. They're all there for a reason. They're all there because they have genes encoding for them. They all serve purposes. Your body knows what it's doing. It does it really, really well. When does your body get thrown off track? It gets thrown off track when you cause your body to become chronically inflamed because of dietary habits, exercise habits, lifestyle habits, environmental toxins, etc., etc., etc. These are the things we need to look at, not trying to mess around with your body and what it does normally and naturally. Just finally on that, at the bottom there in blue, blood triglyceride has also been proposed as a valid predictor of heart disease. Again, if that was the case, we'd expect to see the same thing that we would expect to see in the red graph at the top. We don't see it. It's a normal distribution. Doesn't work for me. Let's move on with some actual science here, people. Okay, so what are these statins? Okay. Statins are a class of drugs which are in the class of drugs which are called the 3-hydroxy-3-methylglutarol coenzyme A reductase inhibitors. Wow, that was a bit of a mouthful, wasn't it? What do? They mess around with your body's ability to biosynthesize the cholesterol. Okay? That thing that your body has basically, not your body individually, but the human body has been learning how to do and how to control for billions and billions of years. So in the last 20, 30 years, we've decided we know better than Mother Nature, and we're going to put this chemical in your body that's going to block that enzyme. And what that does is that reliably and repeatedly has been shown to be associated with around about a 40% decrease in the serum cholesterol. The first comment I'd make about that is, please, if you're on statins or if you know someone's on statins, get them to read the entire pamphlet that comes with those statins. And what you'll see there is a disclaimer that says, this product does what it says on the tin. It says it will reduce your cholesterol in your blood, and it will. However, it is not associated with any change in your risk for heart disease. Wow, they know it. And I'm going to show you some of the studies that back that assertion up. But they have to put that there in small print because they have to tell you this drug, while it will have an effect on the cholesterol in your blood, it will not have an effect on your risk of heart disease 1 iota. This thing will not protect your heart from disease. It will not mean that you live 1 day longer. Interesting. Anyway, that said, how does it work? Okay. Don't freak out. This is a chart which shows one small tiny corner of the metabolic pathways that exist in your body. This one is basically the Mevalonate pathway, and what it shows, the main points to take there is that it starts with a basic metabolic intermediary called acetyl coenzyme A at the top left there. Now, acetyl coenzyme A, the source of that in your diet is basically everything you eat. Like it all gets broken down to acetyl coenzyme A before it's either oxidized for energy or used to build up other things. So be it fats, proteins, carbohydrates, whatever it is, it all ends up in acetyl coenzyme A. So what we're saying is your body can and does synthesize cholesterol from anything you eat whatsoever. So that's another argument for saying dietary intervention is not the answer. Anyway, that's the starting point of cholesterol, the acetyl coenzyme A. So let's follow this through, and don't worry about all the intricacies on this chart. I'm going to pick the eyes out of that for you in a minute and tell you the most important stuff on there. But let's see if we can find cholesterol on this chart anywhere. I'll give you a few minutes. Okay, I'm bored. I'll save you some time. It's not there. Where is it? Well, if you follow this chart all the way down, all the way down to near the bottom there, you'll see that there's a steroid biosynthesis pathway circle there in the bottom left-hand quarter of your screen. That actually indicates a whole nother page. So let's go to that page. Where's cholesterol? Oh, there it is. Bottom left-hand corner, all the way down there. And again, there's all the stuff. All those little squares indicate different enzymes which catalyze different reactions which produce different products or different molecules in our body. Now, why do all these enzymes exist? Okay, the answer is because we have genes that encode for them, and the reason we have those genes is because they have been selected for by natural selection over 3.5 to 3.8 billion years. Every single one of those things that occurs in there occurs for a reason. It has a purpose, and some of them are pretty damned important. Now, I'm going to give you, as I said, I'm going to give you a breakdown, a more simplified version of this to have a look at in a minute as to what some of these really important things are everywhere all over this tree. Okay? And that's stuff that comes from the same pathway prior to cholesterol being formed. There's a whole bunch of stuff cholesterol does and is needed for in our body that if we lower the concentration of cholesterol, we're going to interfere with our body's ability to do those things. But there's also a whole bunch of stuff that's kind of on a parallel branch of the tree, if you like, that if we use this drug, it's going to mess around with our ability to do that. And I'll show you why that is in a minute. But here's a third chart. And what this chart is, this is the stuff that's downstream of cholesterol. These are the metabolic endpoints. These are the pathways by which cholesterol is used in our body to do useful stuff. So, nothing important. Whew. Anyway, here's that simplified pathway that I'm looking at. Okay. What we need to understand is that the statin drug, the HMG-CoA reductase inhibitor, works very, very high up on that pathway. Okay, so there's a whole bunch of stuff, both parallel to cholesterol and downstream of cholesterol, that is affected by this drug as well. And they are things like phenylated proteins, dolichols, sterol, heme, ubiquinones, and everything that's associated with cholesterol itself. Among a few examples, I've just picked the eyes out. There are other things as well, but yeah. So, are any of those things really important? Are any of those things, do we need to worry about those things if we're taking this drug? You bet your sweet behind. Let's go through some of them. Phenylated proteins. Okay. These are involved in cell-to-cell communication, normal inflammation processes, normal control of inflammation, chemical signaling. This is like if you knock down your ability to synthesize these things, basically what you're saying is we're knocking down our ability to communicate with each other. If each of us was a cell and the way that we communicate was the internet, it's saying, well, let's all communicate but not use the internet. Might be a good idea actually, but that's for another day. Anyway, in your body, it's a bad idea. It's a really bad idea to mess around with the level of phenylated proteins in your body, and statin drugs will do that. Okay. Dolichols, nothing important. They're just vital to DNA repair when it becomes damaged by various different environmental insults and various things that cause damage to our DNA on a daily basis. Yeah, I wouldn't worry about it. Take those statins. They'll keep your cholesterol down and it'll keep you alive for how long again? Oh, that's right, not 1 day. Okay. Sterols. Yep, cell membrane integrity again there, folks. Are our cells important? Nah, they're just the things that make up our bodies. Let's mess around with our ability to make our cell membranes, because we've got some ridiculous idea about cholesterol causing heart disease when it does not. Okay. Heme, yeah, that's only a protein that's involved in oxygen storage and transport. So, yeah, I wouldn't worry about that. That's not too important either. And I'm being deliberately flippant here, folks, because this stuff pisses me off so much. This drug is a nasty, nasty, nasty piece of work. And there's absolutely no indication for it. It does not improve the life or longevity of anybody that's on it. In fact, it has side effects that are really, really nasty. And the absolute worst one, in my opinion, perhaps, is that it messes around with your ability to produce a thing called coenzyme Q10, which is a ubiquinone which exists in the mitochondria of your body. The mitochondria are vital to cell function in every cell. They are your energy producers. They are actually little beasties that decided to live with us billions and billions of years ago, that have their own DNA. They live inside us, which keeps them happy and protected, and in return they basically produce all our energy. They allow us to exist. They allow us to use oxygen for energy. They allow us to be multicellular organisms. They are the thing that make us possible. And this drug screws around with their function and makes these guys sick. This can lead to muscle damage, muscle breakdown of an irreversible nature, various forms of chronic pain syndromes, weakened muscles that will be more determined, more predisposed, I should say, to injuries, etc., etc., etc. Oh, and by the way, it's not only skeletal muscles I'm talking about that this drug damages. We're also talking about smooth muscles, and again, they're inside your vascular epithelia, and also your heart is a muscle. So the very organ this drug is supposed to protect is actually put at serious risk from this drug. Alright? So that's just the stuff that's kind of parallel, that's also knocked out by the same drug. But also we're going to reduce our cholesterol by taking this drug, which, you know, we need to remember that is going to interfere with the ability of producing these cell membranes that are so vital to our life. It's going to mess around with our sex hormone production. It's going to make us more predisposed to fractures and early menopauses and lowered muscle mass and functional strength, and it's just a nightmare basically. And also it's going to stuff around with our ability to have healthy nerve cells in our brain and in our peripheral nervous system as well. It's going to predispose us to all sorts of things like dementia and Alzheimer's disease and all sorts of carry on there. And also there's this thing called vitamin D, which, as I mentioned earlier, I believe is pretty vital. And the reason it's pretty vital is because of all the tens and possibly hundreds of thousands of different enzymes in your body. Vitamin D is involved in activating fully 1/3 of those and deactivating those if it is not sufficient. Furthermore, bile is pretty important in being able to take in and metabolize fat. So all in all, what we're talking about here is a bunch of very, very significant risks from taking this drug in return for absolutely no improvement in our risk profile whatsoever. Here's a study that backs that up. This one's a 2010 meta-analysis. So if we look down at the data synthesis here at the bottom, we can pick the eyes out of this. They've followed 65,229 participants for a number of years, adding up to an actual sample size of 244,000 person years of follow-up. They obviously had two populations, people that were on statins and people that were not on statins. Now these subjects were all people who were determined as being high risk of cardiovascular disease. Oh, these are high risk people, we must, give them statins. Half of them were on statins, half of them were not on statins. And what we find is, the last sentence here is the key one, there was no statistical evidence, not the last sentence, the one before that. Okay, which basically says that the use of statins in this high-risk primary prevention setting was not associated with a statistically significant reduction in risk. These drugs didn't keep people alive for 1 day longer. Okay? There was no benefit, there are massive risks. To my way of thinking, that means this drug needs to be withdrawn. There is no argument that can justify the existence and availability of this drug or the prescription of this drug to people on the basis of the scientific evidence that's available. Unfortunately, science is for sale, and there are others who would argue against what I'm saying, and they'll use all kind of spin to do that. But what we're looking at here are the actual empirical conclusions made by the scientists with no axis to grind. The last sentence I was alluding to there says also that there was no evidence that there was a huge difference between one study and another. So they have a massive overall finding that says this drug doesn't actually keep people alive one day longer. And it wasn't like some studies found that yes it did and some studies found it didn't. All the studies were pretty much in line with this finding. This drug does not keep people alive one day longer. It does not provide a benefit.